Hello and greetings from the University of South Florida in Tampa. Please note that this session is being recorded. Thank you for joining us for another session of our Global Health Conversation series. I'm Lynette Menezes, and I oversee global programs for USF Health, which consists of the colleges of medicine, nursing, public health, and pharmacy. On behalf of USF Health International and the Global Virus Network, I want to warmly welcome each of you and our many partners across the world. As you all know, the predicted second wave of COVID cases is becoming a reality across many countries in the Northern Hemisphere. Last week, we added nearly 3.4 million confirmed cases, bringing the global total to now more than 47 million cases. As we continue to learn more and more about SARS-CoV-2, aggressive testing, detection, and isolation, combined with surveillance strategies, remain the cornerstone public health strategies to control and eliminate this infection. And today, I am delighted to let you know that we have two experts, Dr. Tom Unnasch and Dr. Kami Kim, who will share their knowledge about current diagnostics and surveillance strategies for COVID-19. At the end of this presentation, you will have plenty of time for questions. Please type in your questions in the chat box during the presentation, and Dr. Kim and Dr. Unnasch will answer as many as they can. So it is my privilege today to introduce our two distinguished speakers. I'm going to begin with Dr. Kami Kim because the way this presentation is scheduled is Dr. Unnasch will go first, followed by Dr. Kim, and then Dr. Unnasch will end the presentation. Dr. Kami Kim is the Senzivani Professor of Medicine and Director of the USF Division of Infectious Disease and International Medicine. Dr. Kim began her research career as a physician scientist studying pathogenesis of malaria when she was still a medical resident at Columbia Presbyterian Medical Center. As an active infectious disease researcher, she initiated a translational research project to understand the pathogenesis of severe malaria infection. This multidisciplinary project, which involves both clinicians and PhD scientists, utilizes both her clinical experience and expertise in molecular and cellular parasitology. Dr. Kim is also the co-editor of the Sanford Guide that is widely used by physicians across the world to treat infectious diseases. Currently, she leads the COVID-19 pandemic response for USF and its affiliated Tampa General Hospital, advising the leadership of both institutions constantly. Since the start of the pandemic response, she has been heavily engaged in several clinical trials. I think we have like 30 clinical trials at USF now, and she's the PI on several of these, among which is she's the PI of the USF TGH study to evaluate 3D printed swabs as an alternative component in viral sample collection kits. Dr. Kim is also the TGH USF site director for the Regeneron-sponsored clinical trial testing the monoclonal antibody cocktail against the SARS-CoV-2 spike protein. This is an experimental drug that everyone knows President Trump received for treatment of COVID-19. Finally, Dr. Kim has been spearheading efforts for the USF TGH COVID-19 clinical biorepository and clinical registry. She, along with Dr. Unnasch and other colleagues in the Global Health Infectious Disease Research Center, have established a BSL-2 and BSL-3 laboratory to study COVID-19. So we are delighted to welcome Dr. Kami Kim to this talk today. Our second speaker is Dr. Tom Unnasch. Dr. Unnasch is Distinguished University Professor and Strategic Lead of Global and Planetary Health within the USF College of Public Health and is also the co-director of the Global Health and Infectious Disease Research Center at USF. He's a recognized leader in the study of vector-borne infectious diseases and is world-renowned for his pioneering work in eliminating onchocerciasis that afflicts some of the poorest people in the world. Dr. Unnasch has published over 275 papers that are cited widely. At USF, his work has been funded by the Carter Foundation, the World Health Organization, the Department of Defense, Gates Foundation, the NIH, the National Science Foundation, and USAID. Due to his leadership, USF was named the WHO Collaborating Center for Onchocerciasis Diagnostics, which is one of the only WHO centers for this work in the world. Currently, Dr. Unnasch is very engaged in USF's response to COVID-19, both within USF and in the Tampa Bay region. He has implemented a surveillance strategy wherein his lab tests environmental samples taken across USF's 3 campuses. This includes surface samples from high-touch points, such as door handles, elevator button, faucets, and snack machines. People who may have come in contact with a particular location with positive results are then encouraged to get tested for COVID-19. Additionally, his lab also performs pool testing on random samples collected weekly to monitor the overall prevalence of SARS-CoV-2 infection on campus. So we are delighted to have our speakers here today, and without further ado, I'm going to have Dr. Tom Unnasch begin his presentation.
Sorry, I was on mute. Thank you very much, Lynn. So I would like to start today by giving a kind of general overview on how we can evaluate test quality and some of the very important metrics that are involved in developing test qualities. And then I'm going to turn it over to Dr. Kim, who will discuss the several different types of tests that are out there and advantages and disadvantages of each. And then she's going to turn it back over to me, and then I'm going to talk a little bit about some of the basics of pool screening and how this may be used to rapidly and inexpensively identify infected individuals or determine prevalences in a population. So may I have the next slide, please? So just over here on the left is sort of a schematic of COVID-19, which kind of shows you the different parts of the virus. And basically what we can do is to use the different parts of the virus to develop different diagnostic tests that we can target to try and figure out whether people have been exposed to or have been infected with SARS-CoV-2, the virus that causes COVID-19. Perhaps the first target that was identified and the first assays that were developed target the nucleic acid genome of the virus using reverse transcriptase polymerase chain reaction, or RT-PCR. This measures the levels of viral RNA that are present in a clinical sample that you collect from an individual. And it's very sensitive, as we'll see, very specific. However, it's very technically complex, and it's fairly expensive. It requires some specialized laboratory equipment to carry out, which makes it a little bit less useful for many tests than some of the other tests that have come along a little bit later. There have been some other tests that are out there involving nucleic acid amplification as well, isothermal amplification processes. These require less material than the RT-PCR does, or not less material, excuse me, less equipment than the RT-PCR does. So they can be set up and run in less well-equipped laboratories. Finally, the third thing that we can look at to try and detect is not just the genome of the virus, but the proteins that are present in the virus that make the virus itself. The spike protein or the hemagglutinin esterase protein or nucleocapsid protein can all be targeted. And these are fairly, as we'll see, straightforward assays to develop, but have some disadvantages as well. Finally, I imagine many of you heard about serological tests or antibody tests. These do not directly measure the presence of the virus, but they measure the host's immune response to the virus. So it's an indirect measure of exposure to the virus and not necessarily direct infection, current infection with the virus. Can I have the next slide, please? So with these three different types of tests that we have out there, the nucleic acid-based tests, the antigen tests, and the antibody tests, how do we decide which one to use? Well, what we really want to do is look at test performance. And in test performance, we really look at generally three different things. First is the sensitivity of the assay, which is how many of the real positives does a test classify as positive. So if you have 100 positives that you test and you end up with 95 positive results in the test, the sensitivity of test is 95 or over 100, or about 95%. Specificity measures how many real negatives does a test classify as negative. So again, if you have a test where you have, let's say, 1,000 individuals and 50 of them are actually testing as positive, how many of those 950 negative people, really negative people in your population, give you a negative result on the test? Are there any false positive tests that are actually occurring out there? And finally, it's really important to consider the limit of detection. This is how many few of the pathogen organisms that you have that you're trying to detect actually need to be present before you get a positive signal. And obviously, this can really vary depending on the test and how the test is designed. Next slide, please. And one of the really important factors here, especially if you're dealing with an infection that's a fairly low prevalence of infection, is what's called the positive predictive value. And this is basically determined by the sensitivity and specificity of the test, primarily the specificity of the test and the rate of the false positives, and the prevalence of the actual infection in the population. And to give you an example of how positive predictive value can be really important, let's say we have a test with 99% specificity, which sounds pretty good. That means that you only have a 1% false positive rate, and you're testing 1,000 people where the disease prevalence is 0.5%. So if you're testing 1,000 people with a prevalence of 0.5%, you're going to have 5 true positives in that population. If your test is 100% sensitive, you'll detect all 5 of those, and the percentage of the number of true positives is going to be 5. However, don't forget we have a 99% specificity, which means that we're going to have a 1% false positive rate. That means we're also going to have 10 false positives in addition to those 5 true positives. So in this case, the positive predictive value is 5 divided by 15, which is the number of true positives over the total number of positives returned by the test, and it's only 33%. So you can see, if you're trying to do individual diagnostics here, in a population where the prevalence is low, specificity is really important, because otherwise most of the positives that you're going to be getting are going to be false positives. Next slide, please. You go ahead. It's your turn now.
Oh, all right. So thank you, Dr. Unnasch, and thank you, Dr. Menezes, for inviting us to speak. So testing is actually pretty complicated for COVID, and this is because there are different phases when tests are helpful or not. So there is the exposure period, the incubation period, and obviously even if someone is brewing COVID, they may or may not initially be positive. The current thinking is that most people will become positive in the day or two before they are develop clinical symptoms. And generally speaking, we use either RT-PCR or antigen tests to detect active infection. So the active infection will be detectable for the first at least week or two of infection. But what's very difficult about diagnostics is that the RT-PCR test, because it's positive for a long time and is very sensitive, can be positive for many, many weeks. And what we've now realized is that most people whose immune system works well are unlikely to be infectious after the first 8 or 9 days. So the first period where a diagnostic would be helpful during active infection is the PCR test and the antigen test. Then as infection proceeds, you will start having that indirect response, the antibody response. Generally, the IgM develops first and then wanes, and then you'll develop IgG, which is also associated with protection. So a number of studies have suggested that the IgG immune response is associated with both clearing of the virus and development of neutralizing antibodies. So therefore, would be a positive sign that you're able to clear the infection. The duration of these antibodies is actually unknown. Next slide. All right, so antibody tests are very useful and they're commonly used. There are two flavors of antibody tests. The quantitative tests, which are the enzyme-linked immunosorbent assay, or ELISA. And then there are qualitative tests. There are lab-based tests. A chemiluminescent test is one that's broadly marketed by several companies. And then the rapid diagnostic test. And so this detection depends on the immune function and of the person who's creating the antibodies, and generally occurs with some variability within a couple of weeks after infection. And the tests, as I alluded to before, can target IgM or IgG. The IgM happens first. The IgG is later infection and is usually long-lasting and lasts for months to years. Next. So ELISA, for those of you who are scientists or trying to measure the actual extent of the antibody response, is very, very valuable test. So it gives you a quantitative readout. Many samples can be analyzed at once. It is less technically complex than RT-PCR, but does require usually special lab equipment. Its limit of detection is usually higher than the rapid diagnostic tests. So the ELISA can give you quantitative results and is generally a more sensitive test. Unfortunately, this test is expensive and it requires the specialized laboratory, so there may be delays in the resulting. This is the test that's actually used when people are actually looking for surrogates of protection, including what's called neutralizing antibodies. There are other tests that can look at neutralizing antibodies that require a BSL-3 laboratory, so really are not available in the clinical realm. But this test is frequently used in specialty laboratories if we're looking for a surrogate of neutralizing antibodies. Next. So there are also rapid diagnostic tests, and these are very similar to pregnancy tests or malaria tests. These are usually specialized small plastic templates that can be widely deployed, and you can use either blood or plasma. A drop of blood is frequently enough. So these are fantastic if they're accurate enough, but as Dr. Unnasch alluded to, they are frequently less sensitive and less specific, so you can actually have both false positives and false negatives. And in the case of COVID, we know that these tests have had a lot of false positives, so that at this point, we probably wouldn't recommend using them. But the advantage of them is the laboratory is not required, and it's a very familiar platform that many sort of clinics are used to using. Next. All right, so I think I've discussed the disadvantages of the antibodies test. The sensitivity and specificity are not well defined. Generally speaking, the tests that are done in specialized laboratories, either the ELISA test or there have been several commercial formats that are automated and quite good, but require that laboratory to administer. There is, depending on the tests, potential for cross-reactivity to other coronaviruses. It's unclear whether mild or asymptomatic infections may produce sufficient antibody to detect the infection. And I think the other thing is this test is useful in some cases, but it's only really reliably positive within 2 or 3 weeks after infection begins. And obviously in the era of a pandemic, we are interested in picking up the infections during the acute phase when people are potentially passing the infection on to other people. And finally, what we don't know, but is very important, is whether antibody levels, what the meaning of antibody levels decaying after a few months is. And for coronaviruses, generally the antibody response is relatively short-lived and does not persist longer than a year or so. Next slide. All right. So another difficulty with the COVID-19 testing is what specimen to gather. As Dr. Menezes alluded to, there are special collection kits, and that has been one of the many things that has been short during this pandemic. The gold standard is something called a nasopharyngeal swab, and on the left of the slide you can see somebody getting a nasopharyngeal swab, and this is because of the type of swab it is generally collected by a health professional. And you can see it's a thin, flexible swab that is passed through the nose to the back of the throat. And because of that, you can imagine that that causes discomfort, coughing, gagging, and so therefore might potentially provide a risk to the healthcare professional who is gathering the sample, and therefore the sample must be collected using appropriate protective equipment or PPE. So this is the gold standard, which is why it's used. However, there are alternatives, including nasal swabs either collected by a healthcare professional or self-collected, oropharyngeal swabs, and saliva. All of those can be, as I said, self-collected. The problem is that those are collection methods that are possibly less reliable or less sensitive than the gold standard nasopharyngeal swab. So you have a trade-off between potentially more accurate and a better sample versus the difficulties of collecting the sample. Next slide. All right. So we're going to talk about the different types of tests. So the antigen test, as Dr. Unnasch alluded to, is one of the diagnostic modalities that's being commonly used for COVID. It is also detecting the virus, protein from the virus, so should be a surrogate of active infection. And it is much less complex. It can be incorporated into these rapid diagnostic platforms similar to those antibody tests that we showed you. It generally is much less expensive to run an antigen test because of the flexibility as far as format. They frequently also can give you results within 15 minutes. There are many diagnostic tests that have been rolled out in these platforms, including influenza tests. But the problem is we know that these tests are less sensitive than RT-PCR. They generally don't have an amplification step. And though the product insert of most of these tests say that they're 80% in real life practical experience, most people have found that the sensitivity is more like 60 to 70%. And FYI, it was this test that was used for screening of all the people who went into the White House for the Supreme Court celebration gathering. And as we know, we had a super spreader cluster at the White House after people had been screened with this test. So you can see that while it's quite a good test, it will not detect all cases. Next slide. So the gold standard is the RT-PCR test. We use the CDC RT-PCR test. There are various other platforms that have been developed by commercial companies. The WHO has a test as well. And this is basically qualitative detection of nucleic acids from SARS-CoV in biological samples. And if you actually run a standard along with the qualitative detection, you can actually have an estimate of the viral load. So anyhow, usually what we do is we do two genes, N1 and N2. We have a human control, the ribonuclease P protein subunit, and then we do all of this in a reverse transcriptase qPCR kit. So it's an all-in-one kit where the RNA is reverse transcribed and then you have the PCR reaction. Next. All right. So for those of you, most of you are probably familiar with RT-PCR, but basically what happens is that you have the strand of the genome, which in this case is RNA. You have a primer that recognizes a gene on the RNA that, using special enzymes, reverse transcriptase, it extends the primer, and then you have the first strand of cDNA. Then you have another primer that anneals to the cDNA, and then what you can do is repeat the process so you have amplification of that genome. So it is a very, very specific process because you have two specific primers to the genome, and it's also very, very sensitive because you can amplify the genome rapidly and specifically. Next slide. And so because of this, the RT-PCR reaction works even when the target is diluted. So here we have a PCR reaction of a positive control of the BEI reference strain that we got from the reference repository, and a control, and we did 8 1-to-5 dilutions per sample and ran a duplicate. And what you can see is that you can dilute these samples 1-to-5 more than 5 times and still detect a result. So that means that PCR, even if you are off by several dilutions, or even if there isn't very much genome in the sample, you can still detect it. And this comes in very much in handy if you're trying to do strategies to detect a small amount of sample, or pooled strategies, which Dr. Unnasch will talk about in a little bit. Next slide. So RT-PCR, it's highly sensitive and highly specific, as I just told you. Because of its amplification steps, it has a low limit of detection, so you can find it with great sensitivity. But as you can see, it's pretty technically complicated, and because it's technically complicated, you need expensive machines, you need expensive reagents, and also this is done in a specialty laboratory with a relatively slow turnaround, usually 4 to 6 hours. Next slide. So you can see that testing is pretty complicated for COVID, and so we have a lot of questions and conundrums that we still are trying to deal with. As I said, most or a lot of people are symptomatic, but we've also realized that there are asymptomatic people who probably can pass on the virus, and also people are probably more infectious in that first day or so as they're about to develop symptoms. So how do we find these people? And then sometimes, as I said, the PCR result can linger and be positive when we don't think people are infectious anymore. The antigen test, it's not really clear how long those will stay positive in somebody who really has a genuine COVID infection. So then we have the question of, who do you test? How often do you test them? And as I said, there's a big trade-off between the sensitivity of the test. The more sensitive tests are done in specialized labs, so they cost a lot more. However, those are also harder to deploy, and so we initially wanted to detect everyone who is potentially positive, but for epidemiologic reasons, is actually really the most important thing? Is it okay to have a test that's good enough that detects all the people who have the highest level of virus load? And these are questions that we are still trying to figure out, and part of the reason why testing is still 8 or 9 months into this pandemic a little bit controversial and an area of continuing controversy. Next slide. So this is Dr. Unnasch's part. He's going to explain one strategy that we've been implementing here at USF.
So thanks very much, Kami. So as Kami mentioned, with the RT-PCR, one of the nice things about it is that you're amplifying your target, which means that you really need very, very little target in your sample to get a positive result. That means that you can dilute the sample back, as Kami showed, quite a bit, Or what you can do is you can just take smaller amounts of each sample and put whole bunches of these together in a pool, and then screen the whole pool. And if you get a positive result, you know that at least one individual was positive in that pool. This is not something that's really new. It was actually used first to screen draftees for venereal diseases during the start of World War II in 1941 and 42, where they were pooling blood samples and looking for antibodies against syphilis and gonorrhea. It's applicable to a variety of different assay formats like the ELISA or, as I said, the PCR or the RT-PCR. And all you really do is you take aliquots of your samples and you pool them together. Then you assay the pools. And if the pool is negative, you then know that everybody in that pool was negative. If you get a positive result, you know that at least one individual or one sample in that pool was positive. But of course, it could be 1, 2, or 3, or perhaps more. Can I have the next slide, please? So there are a couple ways of using this to really increase the efficiency and throughput of screening large numbers of samples and getting right down to the individual level. Some of them are shown here. Over there on the upper left-hand corner, for example, one of the easiest things to do is just to screen pools. And then if you have a positive pool, you go ahead and you take all the individual samples out of that positive pool and you screen them individually. And then you can identify which of the individuals or which combination of individuals in that pool were the ones that gave the positive result. In the lower left-hand corner, there's another way of doing this. If you're screening large size pools, you can screen subpools first and then screen those individuals in the subpools. So for example, if you had a pool of 50 individuals and that turned positive, you can then make 5 subpools of 10 individuals each, screen those, take the one positive pool that was out of those, or one of the more positive pools that were out of those 10 subpools, and screen the individuals in those subpools and then get right down to the individual. Another way of doing this is the so-called checkerboard or matrix approach. In this case, I'm showing a 96-well screening format because we often use 96-well plates. And the idea here is what you would do would be you would be taking your individual samples and putting them into individual wells of this 96-well plate. Then you pool all of the samples in the rows together, which will be 8 rows. And then you pool all of the samples in the columns together, which will be 12 columns. And then you screen the row pools and the column pools. And what you're going to find is that you'll have one row pool that'll turn up positive and one column pool that'll turn up positive. In this example, it's row B and column 4. Then you know that the sample that was in well B4 was the one that was giving the positive result in both the row pools and the column pools. And so you've screened 96 samples here by only using 20 reactions, saving you about 5-fold in the cost. There's been another method that's been published recently for COVID-19, which is shown over all the way on the right-hand side. A very complex method using a combinatorial approach of taking pools of 48 samples that overlap and putting samples into at least 6 pools of 48. And by using a combinatorial approach, you can screen these 48 pools and get an answer down to the individual level basically immediately, similar to what you're doing with the matrix approach, but saving you, again, quite a bit in terms of money and time in screening by using the pool screening approaches. Can I have the next slide, please? So there are some factors to consider when you're deciding on the pool size. One is the technical limits of the assay. So how far down can you really dilute your sample and get a positive result, even if the sample only has a relatively few copies of the virus, for example. So you don't really want to dilute, a sample that only has 10 copies of a virus 100-fold, because if you do, you're not going to be likely to get a result, a positive result. Another big issue with these is that PCR reactions, and RT-PCRs in particular, are susceptible to inhibition. And so the larger the number of individual samples that you're putting into an individual pool, the more biological material is landing in that individual pool. And the more likely it is that you're going to get inhibitors that are going to co-purify with the RNA through the whole process, which will stop the efficient amplification of the RNA and give you a false negative result. The other big issue here is the prevalence of positives in the samples. Pool screening really works best when you have a low prevalence of infection in your population. The higher the prevalence goes, the more likely it is that your pool is going to end up positive. You know, if you have an infection rate of 10%, for example, and your screening pools of 50, what you're going to end up with is almost all of your pools or all of your pools are going to end up positive. And then you're going to have to go back and screen all those individuals, and you're not going to be saving any time or any money. So the higher the prevalence gets, the smaller the pool size has to be. And this, of course, the smaller the pool size is, the less savings that you're going to get in time and money. Can I have the next slide, please? So there's actually a program that's out there on the internet right now where you can actually calculate your optimal pool sizes based upon the prevalence of infection in the population. So if the prevalence is 1 in 1,000, for example, you can go up to 33 individuals in a pool, and that's going to be very efficient. You end up in that way only really doing 0.07 tests per individual, or less than 1 test per every 1/10 of a test per individual, or 1 test for every 10 to 12 individuals. So you're saving a lot of money there over what you're doing if you're testing individually. But you can see as the prevalence increases, the optimal pool size decreases as well. At 1%, the pool size is 11, and now you're doing 0.2 tests per individual, or 1 test for every 5 individuals, still saving money, but not as much as you were before. If you're up to 5%, it goes down to a pool size of 5, and now you're up to almost 1 test per every 2 individuals. And by the time you get up to a prevalence of 20%, the pool size is 3, and you're doing 80% of a test for every individual. So you're not really saving very much money or time by the time you reach prevalence that's pretty high of about 20% or so. Luckily for us, right now, COVID-19, the prevalence of infection in the communities is still pretty low, and it allows us to use this sort of pool screening approach pretty efficiently. Next slide, please. There's another use to the pool screening testing modality, though. In addition to trying doing these 2-level pool screens and getting things down to an individual level, you can use this to really measure true prevalence in a population by screening pools in the random testing thing, and not really have to drive down to the individual level. So this is a 1-step process. You really don't need to do that step and screen all the individuals in a positive pool, because all you're really interested in is trying to get an estimate of the overall prevalence in the population, and not necessarily which individual or individuals are positive at this point in time. So as I said before, the whole key to this is that positive pools always are going to have 1 individual who's positive, or perhaps even more than 1 individual that's positive. So if you have a positive pool, you can't tell if there's 1, 2, or 3 individuals that were in there that were positive. But if you have a negative pool, you can say with certainty that nobody in the negative pool is positive. And then you can use this to really calculate a probability function that takes the frequency of the negative pools, or the total proportion of pools that are totally negative, and the number of individuals in the pool. And then you can use that to calculate the probability that any individual is infected. And then if you extrapolate that to the overall population, you can use that as a measure of prevalence in the overall population. So for example, if you take this and you calculate a probability that any individual has a 1 in 1,000 chance of being infected, and you go out and you would look at, let's say, 100,000 individuals, you would expect to find 100 individuals that were infected, and your prevalence of your population in the population would be 100 over 100,000, or 0.1%. And the optimal pool size for this method actually turns out, for the statistics, is a point where about 50% of your pools are positive. So you can try and adjust your pool sizes to get 50% of the pools positive, and then you save a lot of money and you get a pretty good estimate on what the overall prevalence in the population is. Next slide, please. We've actually developed a program called Pool Screen that allows you to do these probability calculations very simply, just by putting in the number of pools and the number of pools that are positive, and then it calculates based upon the negative pools. And an example is shown here where we used 100 pools of 20 individuals each, so 2,000 individuals were screened in 100 pools, and we ended up with 10 positive pools. And what you see, the estimate of the infection rate in this population is about 5 times 10 to the minus 3, which is about 0.5%. which works out to 10 in 200. It's a little higher than that because there's, of course, always some probability that a positive pool has more than one positive individual in it. And the program also puts out 3 different types of confidence intervals that you can use to try and get an estimate of how confident you are that the actual point prevalence that you calculated, how accurate that really is. And you can see that in this case, the prevalence, the point prevalences range anywhere from 0.2% to about 0.8%, with a point prevalence of 0.5%. Next slide, please. So we've been using this to do these pool screening assays to test random collections of people, about 300 a week across the USF campus. We're using several different flavors of this test for different purposes. The random testing, as I said, we're doing about 300 randomly selected individuals who are on campus every week, faculty, staff, and students, doing saliva tests, taking saliva samples from those individuals and screening those. We're also screening all of our athletes who are active during the fall season here, all the football teams, the cross-country teams, volleyball teams, and various others. About twice a week, we're doing about 300 or so of these each time we run them. So about 600 of these a week. We're doing these in pools of about 5 pools. And these are nasal swabs, which are self-administered, not a nasopharyngeal swab like Kami showed originally, but just a simple nasal swab. And of course, then we're doing also comprehensive testing of the residence halls when there's a positive that's identified, again, using saliva. We're using pool sizes of 5 because that's the size that's been recommended by the FDA. At this point in time, they've issued an emergency use authorization for pool screening with a pool size of 5. So that's what we've decided to use for our tests. Next slide. So this gives you an idea of the up-to-date current prevalence on campus based upon the results that were through, like, I think, Tuesday, which was the last time we screened the random samples. Among faculty and staff and students, the current prevalence of positivity is about 1%, 0.97%, with a 95% confidence interval of 0.5% to 1.5%. Our athletes are doing a little bit better than the population as a whole, with a prevalence of about 0.5%, again with a confidence interval of 0.3 to 0.7%. And of course, when we went into these residence halls where we had found evidence of positivity, we have a much higher prevalence, in this case about 4.7%, with 95% confidence interval of 2 to 7. So you can see that it's sort of mirroring what you might expect on campus, that the general population is not being as careful as the athletes are, because the athletes want to continue to be able to play. And if you have evidence of an infection in a residence hall or a group of people that are close together, your infection rate is quite a bit higher in that population than it is in the individuals as a whole. So we can use this now to track what's going on campus, and we'll start to be able to see if we're actually seeing an uptick and a increase in the number of positives that are on campus and people who are walking on campus, indicating that we may have a second wave that's starting to develop and maybe starting to occur. But so far, thankfully, we've not seen any evidence, at least on the USF campus, of a second wave, though there is evidence of a second wave occurring in Hillsborough County and the surrounding counties as a whole, which indicates that our student population and faculty are doing a pretty good job of social distancing and reducing the spread, at least on campus. And the USF campus may actually be a safer place to be than being out in Hillsborough County as a whole at the moment. So with that, I'd like to close and say thank you very much, and I'll turn it over to Kami if she has any more closing comments.
Okay, so thank you very much. I think this is the opportunity to for you to pose questions to us. There are actually some in the chat box already. So the first one is that some labs are now offering self-collection swab kits, and can we comment on the accuracy of self-collected swabs? If you review the literature, it's a little bit all over the place. There's been a lot of stuff that's been done for influenza, and it looks like for many cases, particularly for prevalence throughout the population, these are a good compromise between what's feasible and knowing what's going on. Tom, would you like to comment on what your experience is with the self-collection since you've done a fair amount on campus?
Use both self-collected saliva samples and self-collected nasal swabs, and they do seem to be pretty effective. We are picking up, positives, 1 or 2 positives in every one of the pool screening assays that we're running on general. And when they go back and they test the individuals, invariably when we have a positive pool, they're finding at least one positive individual in the pool. So it does seem to be working pretty well. I can't really comment, obviously, on the total sensitivity of the assay because we haven't done a comparison of nasopharyngals versus self-administered nasal swabs. But for surveillance, it does seem to be working pretty well.
Okay. All right. So the next question is, what are the challenges in developing rapid tests that could quickly detect cases and clusters in developing countries? That's a very, very difficult question. And I think part of the problem also is the availability of these tests and reagents. But Tom, you have much more experience deploying rapid tests in underserved communities, particularly abroad. So would you like to comment on how we would get those kinds of tests to developing countries? Tom, I think you're still muted.
Sorry, Kami. Yeah, my comment is I think it's actually going to be a little bit easier than people might consider it to be. And the reason for that is that almost every developing country that I've been through in Africa and elsewhere at this point has RT-PCR capabilities because they're doing HIV viral load screening. So you have laboratories that are central laboratories that are familiar with doing RT-PCRs. They have the equipment that's there, the real-time machines. And then it's just a question of modifying the protocols to change the primers, and you could probably run COVID tests pretty quickly. Of course, the problem there is that the costs are really quite high. You know, just for the reagents alone, based upon what we're paying about $10 to $12 per test for the RNA purification and the RT-PCR reagents. So that's not going to be really very economically feasible in a developing country type situation where you're charging, you know, at least $12 for the reagent costs alone, and probably triple that at least for by the time you calculate in your labor costs and everything else. If it turns out that you can screen larger pool sizes, if we can go up above 5 to like 50 or so, I think then it becomes economically much more viable in a developing laboratory situation.
Yes, and I think also I think it's probably going to require some governmental subsidies. I think, you know, that places that have well-developed HIV programs, frequently the Cepheid Gene Expert platform has been rolled out because it's robust and sensitive, you know, in many developing countries. But those, while they're very good and you can train people quite easily in them, they are expensive. So I think it's going to take some commitment on funding agencies to roll out those kinds of platforms. And that of course is above Dr. Unnasch's and my pay grade as far as figuring out how we're going to pay for those kinds of things. Okay, the next question is, there are reports of people with mild or moderate disease that have neutralizing antibodies that have been detected up to 5 months. Will they be able to prevent infection? Well, the real answer is we don't know. However, the encouraging thing about the antibody testing that has been done is that most people do seem to be able to generate an antibody 2 to 3 weeks after infection, most 2 weeks, almost all by 3 weeks. It looks like if you have antibody, if you measure it, you know, using the fancy neutralizing antibody assays, that there usually is neutralizing antibody there. With other coronaviruses, it seems like those antibodies tend to disappear after a year. And what we don't know is how long you're going to be able to protect yourself from infection. And also, does waning antibody mean that you can't actually have some protective response? There is memory and things like that. So it's entirely possible if you're re-challenged again that you would have enough of a response that you would prevent severe disease or not even have symptomatic disease. But this is something that we don't really know. Tom, would you like to comment?
No, I think you've covered it pretty well. The answer is we don't really know yet. And I think the corresponding answer is time will tell. As we get further and further out and people continue to do these sero surveys of the individuals, we'll get an idea of how long the antibody response actually lasts. But of course, the antibody response is not everything. We actually end up with B cell memory. And even when you don't have any antibodies that are around anymore, you still have the cells that are there that can rapidly produce the antibodies if you're exposed to the pathogen again. So there's probably a decent possibility, I think, that even if you don't have any neutralizing antibodies detectable in your serum, if you are exposed to the virus again, your immune response is going to be much more robust and develop much more quickly than it did in the first level of infection. And chances are that you may still get infected, but you may have a much milder course of disease and possibly no disease at all. But again, time is only going to be able to be the thing that's going to allow us to answer those questions.
Okay. Thank you. The next question is from Dr. Olds. In contact tracing, I've seen people who've had contact with a positive case and tested in less than 2 days. I believe this is too soon to detect infection. And then could you comment on the likely time of exposure that we should use with contact tracing? Okay, so this is a very important question. So the incubation period for COVID is about 2 weeks. In reality, most people, if they're going to develop symptoms and detectable virus, will develop it within a week. So our guidance for people who have a bona fide close contact, healthcare workers, things like that, is to test people about 4 or 5 days after whatever their exposure was. So the peak of people getting a positive virus test is about 5 days, and pretty much, I would say close to 90% of people are positive after a week. So I think you're absolutely right. The reason that we recommend 2 weeks is that the tail is pretty long so that people can be positive as long as 2 weeks out. But I think if you're going to be super sure if there is availability of testing, I would use precautions for 2 weeks, test within 4 or 5 days, and then potentially even repeat another test at the end of the period if you're really worried about it. But yes, this is a dilemma, and generally speaking, with what we've done with managing our healthcare workers is we usually test them about day 5 or so if they really need to know.
Yeah, and as Kami said, this is why people are recommending a 14-day quarantine if you've been exposed, because the vast majority, greater than 95% of the people who are going to become infected and get infectious that will occur within the 14 days, and they'll start to develop symptoms. So you'll know whether you've actually had anything or not within 14 days. Most people are not infectious within about 10 or 12 days after they start to develop symptoms, which is why they're recommending the quarantine period for 10 to 12 days, 10 days after you develop symptoms if you tested positive. So it's a little bit shorter developing symptoms because they're counting in that extra 4 days that it may take you to become positive if you were a close contact.
Okay, the next question actually should be addressed by Dr. Unnasch. Can you comment on the use of a pooled testing strategy for countries that have limited access to testing?
Yeah, once again, I think if we are able to push the pool sizes up, this could be something that could be really a useful way of quickly allowing us to reduce the cost and increase the throughput in many of these countries where they have limited access to testing. You know, if you could do 50 samples in a pool, you can basically do 50 times as many people as you can if you're doing individual testing at 1/50th cost. So I think this is something that really can be pushed forward in these sorts of areas, and not even just there, but also in the United States, where we're still facing something of a shortfall in the amount of testing that's available. So yeah, I think this is something that could be really useful worldwide.
Okay, and another question, which I think both of us can answer, but Dr. Unnasch has been very important in advising the county and the state as far as what prevalence and positive testing rates mean. So the next question is a good question. Comment on the use of test positivity in the absence of prevalence studies to measure infection in the community.
My comment is it is a surrogate for measuring prevalence like we're doing with the random testing program at USF, but it's a pretty awful surrogate. The problem is that if you're using test positivity, you're basically using a self-selected population of people who are showing up to go in and get tested. And there are very few, if any, people at random who are just going to be walking down the street one day and walk past the testing facility and say, gee, I think it'd be a great idea to get tested, and walk in and get tested. Most people who are getting tested for a reason. They either are symptomatic, they're feeling kind of crappy, or they have known that they're in close contact with somebody and they're concerned that they may be developing an infection. And so you're with a self-selected population like that, your test positivity rate's obviously going to be much higher than it is in the random population as a whole, because people who aren't sick and don't think they've been exposed aren't going to show up to get tested. And I think this is particularly important as we move into the autumn period here, because I will predict that the rate of test positivity is going to drop fairly dramatically as we move into the flu and the cold season, because people are going to get the influenza, or they're going to get a cold, and they're going to show up to get tested because they're concerned. And so you're going to have a lot more negatives out there than you would during the summer, when there were very few colds and very few influenza. And so you didn't have this confounding factor in there. So it's a measure, but it's not a very good measure, in my opinion.
And I think what it does measure is whether you're testing enough. So my friends in New York, who are in the emergency rooms in the hospitals, people would show up in the emergency room with something that sounded like COVID, and if they weren't so sick, they would be sent home. And for many of these hospitals that had limited testing, their rate of positivity was like 80%, which is horrifying. I mean, that means that pretty much everybody was having a positive test, which means that you should have a lot of negatives. And the same thing for us at Tampa General. We have excellent testing capacity and an outstanding lab. And at the peak, self-selected people were coming in, hospitalized patients were coming in, and our rate of test positivity was close to 25%. So what that meant is there weren't enough tests in the community, because we know that the population prevalence was not 25%. So it does tell you something, but maybe not entirely what people are interpreting the rate of positivity to mean. So anyhow, the next question is one again for Dr. Unnasch. Some countries are using environmental testing in wastewater surveillance. How effective do you think that is as a surveillance strategy?
It actually looks like it's surprisingly effective. In many of the studies that I have seen, you're starting to see evidence of virus, again, using an RT-PCR test in sewage waste several days before symptomatic cases start popping up in the population. This was first demonstrated in Amsterdam, and it's been demonstrated in several places around the country now. And several campuses, university campuses, are now using this to try and monitor infections in their buildings and their residence halls. I think it's a pretty effective way to go. The disadvantage to that is that the sample prep on the wastewater, because it contains a lot of nasty stuff, is you have to do it under biosafety conditions, and it's a pretty complicated process to undergo. So it's a more complex sample processing, so you can't process as many samples as you could using sort of other strategies as well. But it does seem to be pretty effective. We've actually considered going to that on the USF campus, and talked about that a little bit. We're using a system where we're doing environmental sampling right now, using swabs, as Kami said, of frequently touched surfaces, which is also picking up positives at a fairly high clip. And the frequently touched surfaces those samples are a lot easier to process and handle than poop is. So we're for now sticking with the frequently touched surfaces and haven't moved into wastewater testing.
And I think it's really important. We don't have the deep pockets to test everybody regularly, and some campuses have been doing that, but they admit that they're spending millions of dollars a week. And so I think the strategy developed by Dr. Unnasch is a lot less expensive and is giving us high quality data as far as what's going on. So I think it has been a valuable approach using a multi-pronged epidemiologic survey strategy and targeting high-risk populations like the athletes. So actually, what happens when we find a positive case on campus?
What is happening now if we find a positive case on campus is let's say if we find a positive pool, immediately I report those results and we go back and we do individual testing at the Tampa General Morsani Clinic to identify who that individual positive is. Then there is contact tracing that's done, and the individuals who have been exposed to that person are also tested as well. And the person, there are, I think, 3 or 4 quarantine dormitories that have been set up on campus, and the individuals are moved into the quarantine dorms for 10 days, where their food is brought to them, and they're asked not to leave the dormitories, and they're quarantined on campus at this point. And when we've had environmental samples in the dormitories or in the fraternity houses that have turned up positive, these swabs that I talk about, student health swoops in and does individual testing on all the residents in the residence hall or in the fraternity, trying to identify the individuals who are, again, if they turn up positive, are quarantined.
So far, knock wood, that strategy has been pretty effective as far as on-campus detection of cases. The last question is there a possibility of false positives to the rapid antigen tests? Absolutely. So the antigen tests are protein tests, and they are less sensitive just because of the lack of amplification, and there's definitely a possibility of false positives. We found that in our own analysis. So some of our clinics use the antigen test as a screening test, and we have picked up some false positives. And then I think the most notorious example is the governor of Ohio, Governor DeWine, went to the White House. He'd been being tested regularly. He got screened with an antigen test, was positive, was told he couldn't go into the White House, and then he had confirmatory nucleic acid test that showed that he was negative. So the antigen tests are pretty good, but they tend to have more false positives as well as real positives.
And false negatives, as the Trump White House demonstrated.
Yes, and all of the point of care tests have a significant false negative rate. And the question and controversy is, well, can you make up for it by using those tests more frequently, so eventually you'll find those people? And this is a fairly active argument within the public health community as to what's the best strategy for population-based understanding of prevalence and surveillance. So I think that's the final question. So,
yes,
Dr. Menezes, do you want to?
Yes.
Session?
Yes. Thank you. Thank you so much, Dr. Kim and Dr. Unnasch, for this excellent presentation. I think we learned so much from all the expertise that you shared with us. And for many of our partners across the world, this will be a new way of thinking for some new strategies in which they could probably deliver testing in a more appropriate and timely manner. And also for answering all those questions and sharing some of the newer ways in which we could probably have a better control on this infection. So once again, thank you. And thank you to all of our audience for participating and for asking those wonderful questions. Have a lovely day. Bye-bye.
Thank you for asking.
Thanks a lot.
Bye-bye.